Alright guys, so here is our mini lesson on cells. Now, first thing I want you to think about is the type of cells we're looking at. So there's really three things that you should be looking for. First of all, and probably the easiest to observe, would be the shape of the cell. So when I'm looking at this cell, I notice that it's pretty uneven around the edges. Lots of wiggly lines, not really any clear-cut shapes. That makes me lean towards an animal cell. If we had a plant cell, we would have rigid, straight walls. It would either be box-like, or we'd have multiple sides, but they would be flat. Second thing that we can look for are the chloroplasts. Those kind of resemble our mitochondria, which would be right here. The difference is that if you have a colored diagram, the chloroplasts are always green, and they have really organized stacks inside of them. So I really don't see anything like that on this diagram. The last thing we look for is the central vacuole. That would be a large, almost blob shape that would be right in the middle of the cell. Now, it's not the same thing as the nucleus. Okay, our nucleus is going to be over here. I'm going to label it with an N. Now, you can tell the nucleus apart from the central vacuole because the nucleus will usually be represented three-dimensionally. They'll give it some height. Also, the nucleus has a center called the nucleolus, which I'm going to circle in red. Okay. That's where the DNA is actually stored, in the nucleus. A central vacuole isn't going to have any extra structures inside of it. Its only function is to hold water. So, based on those three things, I don't see flat edges, I don't see any chloroplasts, and I don't see a central vacuole. So, we're going to say that this would be an animal cell. Now, the next thing that we need to do is determine whether this cell is prokaryotic or eukaryotic. So, with those two options, we have a couple ways to remember that. First one, prokaryotic. Pro rhymes with no. So a prokaryotic cell would have no nucleus. Its DNA would be very unorganized, almost like spaghetti, and it would be kind of distributed throughout the whole cell. With a eukaryotic cell, eukaryotic cells have a nucleus. Now, if you can remember that your cells had a nucleus, for example, when we looked at your cheek cells under the microscope, each of your cheek cells had a nucleus in it. You have a nucleus. You are eukaryotic. You can use either one of those to remember the difference between prokaryotic and eukaryotic. So we had already identified our nucleus in this diagram. It'll always be at least fairly centered. It's usually going to show the nucleolus, which we circled in red, and it's always going to be surrounded by this kind of wall-like structure. So this cell has a nucleus. So would it be eukaryotic or prokaryotic? It would be eukaryotic. Excellent. Now let's clean up a little bit around our diagram. The next thing we need to discuss are the organelles. This is where a lot of people struggle. So we're going to talk about each of the major organelles in an animal cell first, since we have our animal cell diagram all up and ready. So, let's see, I used some different colors for our different organelles. First organelle that we'll talk about, since we've already mentioned it a few times, is the nucleus. Now, this year we are studying plant cells and animal cells. So, those are all going to have a nucleus. There really aren't any prokaryotic plants or animals. Prokaryotic organisms would be things like bacteria, and really that's pretty much it. So, we've labeled our nucleus. Now, the job of the nucleus is kind of to function as the control center of the whole cell. If the cell had a brain, this would be it. The nucleus stores the DNA. And that's important because, like we're going to learn in the next chapter, DNA has all of the instructions for living things. The next organelle that we're going to identify would be the ribosomes. Okay, ribosomes will use red. Now, ribosomes can be in a few different places in the cell. The first place we're going to look for ribosomes are on the endoplasmic reticulum. Okay, it's all these little dots, and my finger is not precise enough to label one tiny specific dot, but all those polka dots along that pink structure would be ribosomes. Now, in a minute, we're going to talk about the ER, and that's what makes the rough ER rough, is it's covered in these ribosome structures. They can also be found throughout the cell, so I'm going to put a few dots in the cytoplasm of our cell. The ribosomes can also be free-floating. Now, the job of the ribosomes, their function is to make proteins, which really doesn't sound all of that important until you realize that proteins are required for transportation inside and outside the cell. They're required to help copy DNA. Proteins are essential for life. So these tiny little ribosomes have a huge job. They function in making those proteins that all cells need for life. Alright, the next organelle that we're going to talk about is the Golgi apparatus. This can also be called the Golgi bodies or the Golgi complex. Same basic idea. So the Golgi apparatus kind of looks like a bunch of stuff folded on itself over and over again, almost like an accordion. So I'm going to write this, the Golgi apparatus. There we go. Now, the Golgi apparatus can be anywhere in the cell. A lot of times you're going to find it close to that endoplasmic reticulum. And the reason for that is because the Golgi apparatus's job is to package proteins. That will help to package materials made in the cell to get them ready for transportation. So that Golgi apparatus is close to the endoplasmic reticulum because they have very similar jobs. So when you think Golgi apparatus, think the Postal Service. Okay, they package anything in the cell and prepare it for transport. Now, the transport itself, we'll use green. That would be our endoplasmic reticulum that we've already talked about a few times. Okay, so I'll use the green arrow, and I'm just going to abbreviate on here because goodness knows I wouldn't be able to fit the whole thing. But that stands for endoplasmic reticulum, or we can call it the ER. The ER's function is to help transport materials out of the cell. A lot of times people think that its function is almost a wall to protect the nucleus, but really its key function is that transportation, and that's why it's that folded structure again, just like the Golgi apparatus. Now, there are technically two portions to the ER. There's the rough ER and the smooth ER. The difference would be that the rough ER is covered in those ribosomes. So this diagram really focuses on the rough ER. The smooth ER would also be around the nucleus. When you're looking for the ER, it's very easy to confuse it with the Golgi apparatus. They have very similar structures. So the key is look for the nucleus. The ER will always be wrapped around the nucleus, whereas the Golgi apparatus could be free-floating. Okay, next we're going to label our vacuole. Now, remember, this is an animal cell. In a plant cell, you may not have small vacuoles around the cell. But in an animal cell, we're going to have sporadic vacuoles, and really their function is storage. They could store nutrients, they could store small amounts of water, or they could store waste products. So they're kind of the all-around storage compartment. And really they don't have any special characteristics. They're not particularly large like they would be in a plant cell. They would look similar to a lysosome. Okay, so in this diagram we're calling that green structure the vacuole. You could just as easily call the maroon structure next to it a vacuole. The key is it's a round structure used for storage, which is super important. Now we'll get to the central vacuole in a few minutes. But it's important to remember there is a difference between a vacuole and a central vacuole. Okay, let's see, we gotta find some new colors. Let's use this nice orange. Alright, or teal. Teal works too. So Next, we're going to label our lysosome, which is right next to the vacuole in this diagram. For any of these structures that are kind of sporadically placed throughout the cell, they really are free-floating, so they could be any position in the cell. So this one is going to be our lysosome. Sorry, I'm getting a little sloppy over here. The lysosome is kind of the garbage man of the cell. Its job is to digest waste, and it also can function to digest invaders in the cell. When you think about byproducts of reactions that need to be disposed of, old cell parts, non-functioning organelles, if there's toxins in the cell, all of those would be the job of the lysosome, and that would help to break those wastes down so that the basic parts can be used again. It's almost like a recycling plant. This again is very important. If you have a toxin enter the cell, the lysosome can break it down using enzymes. One of the dangers of the lysosome is that those enzymes, those structures that break things down, they can malfunction. So you could end up, if a lysosome becomes damaged, those enzymes could start to break down living portions of the cell that help keep it functioning. So lysosomes are really important, but again, when something goes wrong, that can be detrimental to the cell. Alright, we have 2 organelles left to label in our animal cell. The first one, let's use this orange color. It's the one that kind of looks like a pill capsule to me. Some people say it looks like a bean. This is the mitochondria. Another long one. Alright, the mitochondria are forever known as the powerhouse of the cell. That's kind of their main slogan. And the reason for that is because the mitochondria are responsible for producing energy. They can break down certain compounds in the cell to get usable energy. This is something that we study in a lot of detail in the next chapters to come. They go through this process that's called cell respiration. So these mitochondria take a basic sugar, and with a few other ingredients, they're able to break that sugar apart chemically to release the energy that's stored in those bonds. So that's really important. Without the mitochondria, the cell wouldn't have energy to function. None of the other organelles would be able to work without this energy. We're going to learn in the next chapter, it's called ATP. And you can kind of think of it as a form of currency, or like money in the cell. Everything has an expense to it. So without enough ATP from the mitochondria, you can't have these transactions or processes going on. Alright, our last organelle that we're going to look at today is one that the function may not make complete sense to you yet in this chapter. But in the next chapter, when we study cell division, everything's going to kind of click with this organelle. And this is called the centriole. Okay, so I'm going to circle it in this dark blue color. And I'm going to have to kind of make a mess of my diagram. I'm going to draw it right over here. So these are called the centrioles. Now their job, they kind of help the cell arrange itself during cell division. Which again, we haven't really talked much about. But cell division is how a cell would duplicate itself, how the cell reproduces. If you start with one cell, well, in the end, that cell is actually going to split in half and make 2 new cells. So you really want whatever you had in this cell to translate over into both new cells. If it's a stomach cell, then it's going to need to be able to function just like the original cells do. And we won't really get into it. I was going to explain a little bit more. We'll save that for the next chapter. But these centrioles, what their job in the cell is as the cell prepares to divide, these centrioles are going to kind of act as the puppet masters. They have these really neat features called spindle fibers, and they use something called motor proteins to help almost pull and rearrange all of the organelles in the cell. And even when we look at chromosomes, which are made of DNA, they'll move that around the cell so that these new cells have all of the information that we need. It's not that one new cell has double of everything. The centrioles help to organize as the cell splits in half. So again, super important. And hopefully that's what this chapter has reinforced to you, that each of the organelles in the cell are crucial for the cell to be able to function. There's none that are really just there for extra. All of these organelles have a job in the cell. All right, the last thing we need to look at in our animal cell before we jump over to a plant cell are just kind of a reminder of the basic features of a cell. The first one is our cell membrane. The cell membrane is this border that goes around the entire cell. Okay, that would be, we've used the analogy of a water balloon. If we have a water balloon, the balloon itself is what holds the water inside. That would be like our cell membrane. It's flexible. It does allow things in and out. It regulates what can enter and exit the cell based on what the cell needs. So that is our cell membrane. All cells have to have a cell membrane. And then the last structure that all cells also have would be the cytoplasm. And we said if our cell was just kind of a blob, that this cytoplasm would be like the gel that kind of holds everything in there. Now we know there's a lot more to it than that, but this cytoplasm is kind of where everything is stored at a basic level. And I'm shading it in and making it look like a mess. I apologize. But that would be our cytoplasm. It's what's holding all of our organelles inside. Again, if we go back to the water balloon analogy, this would be the water inside the balloon. So there you have it. There's our animal cell. Now the last thing we need to look at are the three important structures found in plant cells. Alright, so here's our second cell. Now, the benefit to having a colored diagram is normally if something is a plant cell, they're going to color it green, which is great until you realize that we can't print in color at our school. So you can't only rely on the color. Also, in a real-life situation, if you're working with a basic microscope, you may not have the ability to see in color. Now, here we will look at some plant leaves under a microscope, and we're going to talk about the reason that they are green in real life without having to use a stain. But I don't want to jump ahead quite yet. So if we look at our cell, now I've already given away that it's a plant cell, but if I hadn't mentioned that, we've got to think back to those three things we look for. First thing is the shape. Now, again, this is a cartoon, but if we look at the shape of the cell, it does have some flat ridges to it. It's not a perfect square, but there's definite flat boundaries in portions of the cell. Now, that being said, I'm not comfortable with calling this a box, so let's go to our second requirement for a plant cell. We're going to look for something called the chloroplast. Now again, this diagram is labeled, which is great. That won't always be the case. So we said the chloroplast kind of looks like a circle with a bunch of really organized stacks inside. So, well, now I see those capsule-shaped structures. I've circled one with the black. Those were the mitochondria. They kind of look like chloroplasts, but they're more squiggles than stacks. Now, right above that, and let me circle it in red, I see something that might pass for a chloroplast. It's definitely green, which is our first sign, but it looks like if we could zoom in, it has some really organized stacks inside of it. That looks like a chloroplast to me. But again, maybe we're not 100% sure, so let's check for our third indicator, that really large central vacuole. Now we said that organelle's job is to store extra water in a plant. Well, when I look at this cell, this is, let me circle in orange. Here's our nucleus. That's clearly the nucleus. It has that nice dot in the middle for the nucleolus, but right next to it, let me change colors. We'll go purple. This big circle right here, that's huge. Look at the proportion of that compared to the cell itself. That's definitely our central vacuole. that's what stores extra water in the plant cell. We talked about in class how if a plant doesn't have enough water, that central vacuole will start to contract, and that can actually cause the plant to shrivel up. So, we said we weren't sure about the shape of our cell, but we found a chloroplast, and we definitely have a central vacuole. That means that this is definitely a. Now that we've established that we know this is a plant cell, we just need to make sure that we know all of the additional organelle functions. So I've already listed them, so we're going to go kind of quickly. Now, the first one would be our cell wall. This diagram is labeled already. So the cell wall, we've got labeled right here. I circled it in that faint green color. The cell wall is located outside of the cell membrane. So it would be that very exterior portion. Now, the job of the cell wall is different than that of the cell membrane. The cell wall's job is to provide a rigid structure to the cell. So the cell membrane's function was to control what enters and exits the cell. The cell wall is really more of a structural support. It certainly does help protect the cell from things coming in and out, but it also gives it that nice firm structure. So that would be our cell wall. That is only found in plant cells, maybe some algae, and a few other species, but primarily plants. Our second structure is very important to the plant. I'm going to use red. That would be the chloroplast. Okay, so we've already described them. They're that structure that is kind of oblong, like a squish circle, but it has really organized stacks inside of it. Now, the chloroplast's job is to capture sunlight, which sounds kind of important, but really this is crucial for the plant to survive. The chloroplast has something called pigments inside of it that we'll study in the next chapter. And those pigments help absorb light instead of just reflecting it back. Once the chloroplast has trapped that sunlight, it can't just store it as sunlight. Sunlight has a crazy amount of energy to it. It is energy. So what has to happen is there's a process in the chloroplast that transforms that sunlight into stored energy. This is called photosynthesis. In the end of photosynthesis, we have energy stored in sugar. Now, if you remember back to when we talked about our mitochondria, mitochondria's job is to take a sugar and break it down to give a usable form of energy called ATP. So in a plant cell, the chloroplasts and mitochondria work hand in hand. The chloroplast's job is to trap the energy and store it, and then once the plant cell needs more energy, the mitochondria is able to break down that sugar and get the usable energy that we called ATP. Our last structure found in the plant cell that we need to talk about is the central vacuole, which again we talked about a little bit already. Now, when we talked about an animal cell, we said that the vacuole's job was for basic storage. So if you can remember that vacuoles store things, that'll start you down the right track. In plants, the central vacuole is really kind of designated to storing water. And when we look at photosynthesis and respiration, we're going to realize how important water is. So this central vacuole will store that extra water to help provide for the plant when maybe there isn't rain after a few days, which we've certainly experienced this summer. Now, that central vacuole does have a secondary job. When it's really full, that's actually going to help support the plant also. Think of a really nicely watered flower. That stem is going to be nice and firm and crisp, whereas if you stop watering a plant, one of the first things that it will do is wilt. And that's because as this central vacuole shrinks, well, think about it, if it shrinks down to that size, well, now there's a lot more room in the cell. The cell itself will start to shrink a little bit. So that central vacuole's primary job is to store water, but one of the benefits of it is that it will help support the plant cell. That's really everything that you'll need to know about cell organelles this chapter. So we talked about the difference between plant and animal cells. So again, the three things that we're looking for are the shape, we're looking for chloroplasts, and we're looking for that really big central vacuole. Now, we also mentioned the difference between prokaryotic and eukaryotic. Okay, we said think no. Prokaryotic does not have a nucleus, whereas eukaryotic, you have a nucleus. You are eukaryotic. And then really the last thing that we focused on was the function of the organelles. Now, if you can remember all of that, you're going to be golden for pretty much the remainder of the year. Cells are the building block of all living things. So even if you're not particularly interested in learning about how a cell functions, realize this affects every component of life. Everything you do is a function of a group of your cells working together. So hopefully this helped you study. Hopefully maybe you found it at least a little bit interesting. And if not, hopefully at least learn something.